My name's Adam Haber. I'm sorry, I believe there are some technical difficulties, so I'm glad that we can get started. I'm glad to be here for Asthma Grand Rounds, and um, I wanted to sort of jump. I wanted to provide the whole talk on this first slide to give an overview of what I'm going to talk about. Um, but I guess I should introduce myself. So I'm an assistant professor at HSPH at the Chan School of Public Health in Environmental Health Department. And my program is called Molecular and Integrative Physiological Sciences. And I'll talk about this a little bit more, but we use a molecular lens to understand environmentally mediated disease. In particular, my lab is focused on asthma. So what I'm going to talk about today is I'll provide some background on the role of the mucosal epithelium and how it's senses the environment and how, as I'm sure many of you know, this can contribute to asthma. I'll talk a lot about heterogeneity of airway epithelial cells and how this provides a division of labor with specialized subtypes performing biological functions. I'll give an overview of some of my previous work showing that discovering new types of rare epithelial cells and identifying their functional roles in maintaining homeostasis in the airway. Two of these subtypes I'm going to talk about a lot are tuft cells, which can act as sort of alarms or sensors, which sense some threat and then trigger inflammation, trigger immunological cascades, and ionocytes, which were recently described by myself and others starting for 2018, and they play really important roles in regulating airway surface liquid. In the second part of the talk, I'll talk about some unpublished data from my group in collaboration with Laura Bankova's lab at Brigham and Women's Hospital, using single-cell sequencing to characterize heterogeneity of epithelial cells, particularly in the nasal mucosa. So I'll try to convince you that if you wanted to have a prime site for monitoring everything coming into the lungs, the nose would be a good such site. And I'll show that our analysis identified that there's surprisingly many of these rare of epithelial subtypes which are rare elsewhere in the lungs, but in the nose, in the mouse nose at least, relatively common. And I'll make some comparisons to human and show that in the nose, tuft cells actually have a few subtypes. They're heterogeneous. And we'll look at how the nasal mucosa responds to inhaled allergens in a mouse model of asthma. That's a mold allergen. And look at contributions of tuft cells to tissue modeling and implications for allergic disease. So this is going to be an overview for what I'm going to talk about over the next 45 minutes. So as I mentioned, I'm an assistant professor in MIPS, which is Molecular and Integrative Physiological Sciences, and I'm actually a computer scientist. And so a lot of the work I'm going to present is sort of data analysis, sort of computational biology. But in the environmental health department, we really focus on understanding how exposures can drive toxicity and disease pathogenesis. So I'll focus here on how my group applies computational biology to understand mucosal barrier tissues, and particularly the epithelium of the airways of the lungs. Let me see if I can grab a tool to hopefully you can see that. And so mucosal barrier tissues are really where the rubber meets the road in terms of the environment and the body. So if we understand how these tissues work, we can understand how environmentally diseases like asthma, we can understand their pathophysiology. So what does mucosal barrier tissue have to do? So this is a schematic which is intended to try to convey some of the cellular complexity that's at play in mucosal barrier tissues. So this is a depiction of the intestinal epithelium, but most of this is true for other mucosal tissues, including the airway. So this is the sort of villus protruding into the intestinal lumen or the environment, and this is the crypt. But most importantly, this is the epithelium. So these are the wall of cells, which the main function of which is to prevent entry of noxious substances and microbes into the body. But they also serve many other functions. They can directly mediate antimicrobial defense by producing antimicrobial peptides. And importantly, they can regulate the local immune system through antigen presentation, production of cytokines, and other inflammatory mediators. And they have to mediate tissue repair in the instance of damage, And perhaps most importantly, they're really important in sensing the pathogens that are in the local milieu and directing whether there's an immunological response or tolerance. And so what's really important, I think, for this talk is that this labor is not equally distributed across the epithelium, but these functions are actually carried out by specific subtypes of epithelial cells. And so in understanding the specialized character of each of these subtypes, that can give us clues about overall tissue function and pathogenesis of specific diseases. So goblet cells are actually implicated in a lot of mucosal diseases, but for example, inflammatory bowel disease, different types of infection, ciliated cells, of course. Similarly, as I mentioned, tissue repair has to be mediated typically by a local pool of stem cells. In the gut, they reside in crypts, and in the airway, I'll talk more about where the stem cells are. Antimicrobial defense is mediated by Paneth cells in the gut, which produce a range of peptides, which are actually called defensins. And in the airway, these club cells, which are rendered a lot like avocados here, perform a sort of analogous role. And two of the subtypes here that are implicated in immune cell activation and thus in inflammatory diseases like asthma are the tuft cell, which I'm really going to focus on, but also the pulmonary neuroendocrine cell, which I'll talk a bit less about. So if you're in mucosal tissue, you really need to work out what's going on in the outside world. And so I like to think about this as a sort of information maximization problem. And so how does this tissue maximize the information that it can learn about from the outside world? And so I think a sort of natural approach is to deploy sensory units in a manner which is spatially rare. So if you wanted to sense information across the whole ocean, you don't put all the arrays in one place, you sort of distribute them sparsely across a large area. And similarly for like the Great Wall of China, for example, the sentry towers are sparse. And that's what we tend to see with epithelial cells, is that there are sort of rare ones which play a particularly important role in sensing. So how can we kind of map out this complexity? And since I did my postdoc in Aviv Regev's group at the Broad Institute, we've been using single cell RNA sequencing to sort of map out this complexity of epithelial cells. And we've begun with the intestinal epithelium. And in collaboration with Moshe Biton, who's now faculty at the Weizmann Institute in Israel, we developed this strategy, sort of isolate epithelial cells by FACS sorting on EpCAM, and then performing 3' droplet-based single cell sequencing. And there's a number of ways to do this, but typically we use the 10x chromium. So this was data that was published in 2017. We mapped out the intestinal epithelial cells using single cell seq. On the left is a dimensionality reduction visualization of what we were able to find. And so each of the little dots here is a single cell, and they're colored by their assignment to clusters using an unsupervised clustering algorithm. And we can then interpret those clusters and map them onto some of the known diversity of intestinal epithelial cells. So this cloud of red points are goblet cells. There's lots of stem cells and progenitors, which give rise to enterocytes. But for this talk, what was most interesting to us was this rare population of tuft cells depicted in orange. And we were able to then use differential expression tests to identify the molecular transcripts which were specific to each subtype of epithelial cell. So here, each column is a single cell, and each row is one of these specific marker genes. And we can see that they display this cell type specific pattern, and we could identify transcripts that were specific to tuft cells. So we profiled all of these in detail and looking for clues to all of their functions. But for my talk today, I'm really going to focus on tuft cells. They are a relatively recently characterized subset of epithelial cells. They're named for they have a tuft of microvilli, which is visible using an electron microscope, although I'm not showing you that, but that you can take my word for it. But they were sort of a curiosity from the 1950s when they were discovered up until 2016, when these 3 high-profile papers identified that they actually sense the presence of H. polygyrus, which is a parasitic worm, and secrete cytokine IL-25, which can induce proliferation and activation of tissue-resident innate lymphoid cells, which then produce IL-13, which acts on the intestinal stem cell to drive the production of goblet cells. So this is sort of homeostasis on the left. On the right, we have goblet cell metaplasia and also tuft cell metaplasia. So there's this remodeling of the intestinal epithelium that's necessary to expel the worm. And this is the sort of canonically called weep and sweep. So lots of mucus production from goblet cells, and the proliferation of the epithelial stem cell leads to a sort of escalator of epithelial cells up to the top of the villus, which can actually dislodge the worm. So linking tuft cells to this well-known type 2 immunological response really emphasized how sort of profoundly important they can be for mucosal immunology, even though they're very, very rare, only 1% of the intestinal epithelium at homeostasis. So we wanted to see what we could identify from our tuft cells in the intestinal epithelium. We had 140 of them. And then we sort of reran our unsupervised analysis, and we're able to identify that about half of them were sort of immature on their way from differentiating from a stem cell to a tuft cell, but 2 of them appeared to be fully postmitotic and nonetheless distinct. And we ran differential expression testing to identify the molecules that were specific to each. And sort of surprisingly, we identified that CD45, which is commonly thought of as a hematopoietic marker or an immune cell marker, was expressed by a subset of these tuft cells, which we're able to validate using single molecule FISH. The CD45 in green is co-localized with the tuft cell marker DCLK1 at the mRNA level, but also at the protein level using immunohistochemistry and flow sorting. The other thing we were excited to see was, given the role of these cells in recruitment and activation of local immune cells, we wanted to look at their cytokine profile. We found that both of these subtypes that we had identified expressed IL-25 as expected, given those papers I was just talking about, but only one of them expressed TSLP, which was exciting. As many of you will likely know, TSLP is a very important type 2 cytokine that can activate a mucosal immune system. This was also validated using single molecule FISH. So now turning to the airway epithelium, which I imagine would be of more interest to this audience. We wondered what type of epithelial cell heterogeneity was present. When we began this study back in 2018, it was sort of thought that there was 3 types of airway epithelial cells: a basal stem cell, which gives rise to this club cell intermediate, which differentiates into a ciliated cell, which performs mucociliary clearance, sort of the workhorse cell of the airway. But there were also reports of other types of airway epithelial cells, which perhaps a little less characterized. Certainly the brush cell, which we now think is very much analogous to the intestinal tuft cell, and the pulmonary and urinic cell were somewhat less characterized, along with goblet cells. So we then applied droplet-based single-cell-seq to the mouse trachea. This was a collaboration between Aviv Regev's group and Jay Rajagopal's group at MGH when I was a postdoc. And we mapped out, this time we have 7,000 epithelial cells, so you can see the numbers are increasing as this technology matures. But what we saw that was perhaps not surprising was 90% of the data was comprised of these 3 canonical types: basal, club, and ciliated. But then we do see these rare clusters corresponding to some more rare types of epithelial cells, so goblet cells down here, which in the mouse trachea are actually very rare at homeostasis. That's not, of course, true of human lungs, but in mice, there's not very many goblet cells in the mouse trachea. But we also had some tuft cells and neuroendocrine cells. So this was exciting. We wanted to see if the 2 subtypes of tuft cells that we identified in the gut were there as well. It appears that they are. And they were, we validated this, the markers of each subtypes with IHC on the right. And what we saw was that there's this TUF1 subset, which expressed a whole range of taste GPCRs. And on the left, a bunch of molecules that encode enzymes that are involved in the synthesis of eicosanoids, and particularly cystinolycotrienes, alpha-C4 synthase, and FLAP, LX5AP, particularly important for producing these inflammatory eicosanoids. We again saw that IL-25 was specific to tuft cells in the airway. and that there was an enrichment, although relatively weak, for TSLP in mouse tough cells. So after showing that these molecules that were present that were needed for the synthesis of eicosanoids, Laura Bankova's group then was able to show that it's not just that these transcripts are actually present at the mRNA level, but these molecules are actually produced at very high levels in response to an allergen. So on the left, she's showing that at baseline, there's relatively few TCLK1 positive tuft cells, but after inhalation of Alternaria, which is a mold allergen, we see this hyperplasia of tuft cells, which is relatively analogous to what was observed in the gut, which is a significant increase in the number of tuft cells. But also the production, I'm actually not sure now which cystine leukotriene is being labeled, but I believe that this is this production of cystine leukotrienes by tuft cells in response to two types of allergens. So dust mites and Alternaria, both are implicated in asthma exacerbations. So this was really confirming that these tuft cells are actually able to produce inflammatory eicosanoids and not just that they have the mRNA molecules. So another population that I haven't yet mentioned was detected. So we've got basal cells, club cells, ciliated cells, tuft cells, neuroendocrine cells, and then this purple population, which we hadn't yet identified, and it was marked by the transcription factor FOXI1. So in Jay Rajagopal's group, they generated FOXI1 EGFP mouse, and then they could do whole mount stains to observe these cells. And they confirmed what we observed in the single cell data, which was that there is a FOXI1 positive cell, which makes up about 1% of the tracheal epithelium. And then when we looked at the transcriptome of these FOXI1 positive cells, we found that they express markers of a cell type called ionocytes, which are found in the skin of xenopus and zebrafish. We found that in two types of single cell seq. So this is droplet-based and this is plate-based, which has a higher coverage per cell, so we can detect things that we might miss in the droplet-based data. But what was very important was that we found that CFTR, which is cystic fibrosis transmembrane conductance regulator, so this is the gene which, when mutated, can give rise to the inherited disease cystic fibrosis, was very specifically expressed in both single cell seq datasets by these ionocytes. So then we deleted these ionocytes using FOXI1 knockout mice, grew airway epithelial cells in air-liquid interface, and then performed some microbiology experiments and showed that the airway surface liquid was quite different. It was quite a lot thicker, more viscous, and the ciliary beat frequency was dramatically different. It was actually increased, maybe counterintuitive, but we interpreted this as some sort of compensatory mechanism for thicker mucus. And we can also sort of observe this thicker mucus in optical coherence tomography experiments, which were done by Steve Rowe's group in Birmingham. So yeah, I guess just to close that out, so the pulmonary ionocyte was therefore implicated in the pathogenesis of cystic fibrosis, because all of this mucosal pathology are sort of hallmarks of CF disease. So an open question coming out of these studies was, okay, we've started to identify new and rare epithelial types in airway function, but what are they actually doing, not just in homeostasis, but also in the context of inflammation? And the ability of airway tuft cells to produce some of these inflammatory signals, particularly these cystine leukotrienes, but also cytokines, IL-25, TSLP, and other eicosanoids, is starting to implicate them in pathogenesis of inflammatory diseases such as asthma. So at this point, we wanted to look at the nasal epithelium. As I mentioned at the beginning of this talk, nasal epithelium is sort of a prime site to observe the inhaled airstream and detect any inhaled threats, such as allergens or other noxious substances. But less was really known about what was present there in terms of the cellular diversity. So there was this study that used single cell seq, the plate-based technology, which was able to sequence 700 cells in 2017, and they sort of mapped out this trajectory from the olfactory stem cell to the olfactory sensory neurons, or OSNs, which actually mediate olfaction. But a lot of the cellular diversity here was not yet characterized. And in particular, this microvillus subset was relatively enigmatic. So in collaboration with Laura's group, we wanted to apply some of these single cell approaches to see what we could see and see if we could characterize some of this heterogeneity, particularly these enigmatic microvillus cells. And so Laura's group used chat GFP mouse to enrich for the chat expressing subsets of microvillus cells, which also express TRPM5. And so we were able to enrich for these cells, and so they're typically rare, but after enrichment, we get a lot of them. But we were sort of surprised to see that there was also quite a lot of this other subset of microvillus cells, which were TRPM5 negative. So historically in the literature, MVCs, or microvillus cells, are known to have two types, TRPM5 positive and TRPM5 negative. But what the difference is between the subtypes is relatively uncharacterized in terms of their function. And so one of the things we wanted to do is say, okay, what's the relationship of these microvillus cell subsets to some of these rare cells that we've been identifying in the large airways, particularly in the trachea? So we applied a machine learning approach. We trained a random forest classifier we basically gave it the data from the trachea and said, okay, if you can learn a model of all of these epithelial cell types, which would you predict are present in the data from the nose? And what the classifier actually said was that these microvillus cells, which are TRPM5 positive, are exactly like tuft cells and sort of very unambiguously like tuft cells. And the TRPM5 negative subset are exactly like ionocytes. So about 92% of these TRPM5 MVCs were classified as tuft cells, and 96% of the TRPM5 negative MVCs were classified as ionocytes. So this was sort of cool. We were able to sort of show, based on transcriptome-wide measurements, was that these two types of olfactory microvillus cells actually correspond to tuft cells and ionocytes. And what was really surprising was that when we looked at these tuft cells and ionocytes, which are both pretty rare in the trachea, particularly ionocytes, less than 0.5%, in the olfactory epithelium, they're much more common, almost 10 times more common. And so this was sort of surprising, but provocative. So Laura's group was then able to stain for chat EGFP, which was marking the TRPM5 positive subset. So that's the tuft cells. And here they are in the olfactory epithelium. And also staining for CFTR, we could see the ionocytes. So both of these subsets are really present in the nasal mucosa, particularly in the olfactory epithelium. And this was sort of confirmed using Cochlin. Here are some Cochlin, this is another ionocyte marker. So here are some Cochlin positive ionocytes. So we then wanted to know if this was also true in humans. So there had been a previous study published in 2018 with Jose Ordovas Montañez as the first author, who's now faculty at Boston Children's. And they had sort of mapped out the epithelial cells in the respiratory mucosa of human noses, but they hadn't noticed these rare cells. So we went back and reanalyzed the data, and we could confirm that they're sort of very, very rare, but there is a population of what look like microvillus cells. And if we kind of pull them out and analyze them separately, it does look like there's two types. And when we look through differential expression at their markers, we can sort of confirm that they do correspond to ionocytes. So we see CFTR and FOXI1, the two markers that I mentioned, but also other ion transporters like these chloride channels, and this is ENaC, the sodium channel. So these are for sure ionocytes. And the tuft cells were expressing a lot of the markers. We're expecting not all of them, but certainly seeing ALX5, which is important in icosanoid synthesis, the receptor for IL-25, and interestingly not IL-25 itself. But these were pretty clearly tuft cells and ionocytes in human nasal mucosa. Subsequently, there was another study published in Nature Neuroscience from Brad Goldstein's group, and we did the same kind of post hoc analysis of their published data. And we confirmed that, again, there's tuft cells and iridocytes are present. And this data was a bit later, and so there are more cells. And we were able to detect with a bit more coverage that the tuft cells are actually expressing really all of the markers we expect to see, particularly IL-13 receptor, which, as I mentioned, is important for their role in type 2 immunity, and also the transcription factor PRO2F3, which is now recognized to be the sort of canonical lineage factor that specifies tuft cells. So Laura then validated using immunofluorescence that there are DCLK1 positive tuft cells and advilin positive tuft cells. These are interchangeable markers, but confirmed using 2 different antibodies that they're present. And in the olfactory epithelium, we can also see that advilin positive tuft cells are present very close to OMP expressing olfactory sensory neurons and NCAM1 positive neurons. So this is a really beautiful stain showing the sort of cellular diversity of the human olfactory mucosa, but also importantly that these advilin positive tuft cells are really there and not super rare. So we then wanted to see if they were all the same, as we'd observed in the gut and in the airway, that there are actually different types of tuft cells. So we took out our subset of TRPM5 positive MVCs. We subclustered them, identified, in fact, that there are 3 subsets. And what I'll show you over the next few slides is that they're actually defined by not only unique transcriptional profiles, but also by differences in morphology and anatomical location within the nasal mucosa. So the most common subset, I guess I'll begin by saying that all 3 of these subsets express core tuft cell markers. So TRPM5 is a taste transduction channel. LTC4 synthase, as I mentioned before, is a core enzyme involved in eicosanoid synthesis. But interestingly, DCLK1, which has proven to be a really important tuft cell marker in other tissues, in the nose is actually only expressed by 1 subset. So this was a potential gotcha for would-be scholars looking at tuft cells in the nose. It would be careful using DCLK1 as a marker because it's definitely not expressed by all of them. But we were able to identify that neurogranin, which is a novel marker of tuft cells, is sort of specific to some subsets and not others. And G-alpha-gastusin or GNAT3 was expressed only by 1 subset. So in a sort of more what's the word, like can't think of the word, but yeah, if we look at differential expression to identify some of these markers that are specific to each subset, we could identify some of these markers, neurogranin that I was just talking about, and G-alpha-gastusin are specific to these subsets, and that each of these subsets actually have distinct receptor profiles, and there are some adrenergic receptor expression in this SCC subset, which I should probably explain why I'm calling these things that. But really, they're quite divergent in terms of their expression of these markers, and they're specific to different locations. So particularly this subset, which I'm calling tuft SCC, which stands for solitary chemosensory cell, was really enriched in the respiratory epithelium of the nose, much more so than the olfactory epithelium. And we confirmed that both from our single cell data and then by using deconvolution of bulk RNA-seq data and confirming that the tuft SCC subset is really specific to the respiratory olfactory epithelium, whereas the other 2 subsets are detected in both respiratory and olfactory epithelium. And this was sort of surprising because it was known that these sort of pomegranate-shaped tuft cells, which were previously called TRPM5 positive MVCs, exist in the olfactory epithelium, but was not known that they exist in the respiratory epithelium as well. So that's something I sort of glossed over, is that there is this important morphological difference between these round globular tuft cells that are majority olfactory epithelium and these more elongated tuft cells, which appear only in the respiratory epithelium. So this is the sort of morphological difference that I was sort of talking about, and also differences in anatomical location. So these beautiful whole mount stains were done by Laura and her postdoc, Saltanat. And we can really see that these tough cell subtypes have both morphological and anatomical differences in sort of where they're found. So next, we wanted to see how this heterogeneity is actually involved in responding to inhaled allergens. And so we turned to a mouse model of what's often called the mouse model of asthma, but is really a mouse model of allergic inflammation in the airways, which was an inhalation of the mold allergen Alternaria. And then we performed single cell sequencing. And so we're detecting this time with no enrichment. So our brush cell population or our tough cell population is much smaller, and our ionocyte population is certainly there, but we're not seeing as many as we did when we had an enrichment. And this time we have immune cells as well. And the first thing we really noticed when we looked at how this distribution really changed in the presence of the allergen is here we can color this UMAP embedding. Instead of coloring it by cell type at the top, we can color it by density. And we saw that this population here, the stem cell population, really sort of increased in number in the presence of the allergen. So, and we saw a relative decrease in the number of mature neurons, but we really did see this big increase in the stem cell population, which was surprising. So we then wanted to look at whether any of the cell types in our data were clearly proliferating, which could explain this increase in the number of stem cells that we observed. And so this plot is showing the result of a regression model, which was testing for whether there's an increase in the proportion of each cell type. Each cell type is a row. And testing for an increase in the proportion of proliferating cells within each cell type. And we saw that, so here, if anything is along this line, it means there's no change in the proportion of proliferation within the cell type. But we saw that, sorry, this is after Alternaria treatment. We saw that only the stem cell compartment, only the HBC subset showed this really stark increase in the proportion of proliferating cells. And here's another way of visualizing that same data by just saying, what's the proportion of proliferating HBCs inferred from the single cell data? In the olfactory epithelium, it goes from like 2% up to 12 or 13%. So this big increase, a relatively big increase in the proportion of proliferating olfactory stem cells in response to the allergen was really notable. So we then turned to some bulk RNA-seq to get really high coverage data. And we used antibody-based sorting to identify mature sensory neurons, immature sensory neurons, and our HBCs, which were identified using the marker BCAM, recently published by Nora Barrett's group, and studied them in response to different allergens. And the first thing we saw was that most of the transcriptional response that we saw was really in the basal cell compartment. So this plot shows the number of differentially expressed genes in response to both Alternaria, but also DIRP and Aspergillus, two other allergens. But really only the HBCs showed a significant transcriptional response, and the olfactory sensory neurons showed little transcriptional upregulation. So what was in this response is shown in this volcano plot. So on the right-hand side are genes which are upregulated by basal cells in response to Alternaria. And what we really saw prominently was MKI67, which is a marker of cell cycle of proliferation. And so if we colored all of the genes that are involved in cell cycle in red, it was really obvious that a large part of the transcriptional response that we observed in the basal stem cells was actually because they're activating proliferation and they're trying to divide. So then Laura's group stained for Ki67 and confirmed that there's this green staining. Oh, sorry, this is PBS, so this kind of relatively rare green staining. But after Alternaria, we saw this increase in proliferating basal cells. So these are the stem cells of the olfactory epithelium, and they exist along the basement membrane. And so green here is the proliferation marker. We can see these nerve bundles stained for TAJ1 and these olfactory sensory neurons. And OMP is the mature sensory neurons, just to orient you. And then using intracellular facts, we could see that at baseline, there's relatively few keratin 5, so that's the marker for the basal stem cell, and Ki67 positive cells, so these are proliferating basal cells. But after the alternaria treatment, it increases, and we can quantify that. And here's the p-values. And so after 36 or 39 hours, we see the significant increase in the proportion of proliferating basal cells in response to alternaria. So we then wanted to look at our single cell data and see what we could infer in terms of the process of neurogenesis, of differentiation from horizontal basal stem cells into olfactory sensory neurons and the other epithelial cells as well. So we performed what's called pseudotime analysis. And the basic idea is that if biological processes are dynamic, when we do single cell sequencing, we're kind of sampling from them at different stages. And we want to have an algorithm that can reconstruct that sort of pseudo time, that biological time, where the processes are changing. And the process which is most typically studied is differentiation. So in this case, pseudotime is attempting to reorder our single cell transcriptomes along the differentiation trajectory. And so we applied an algorithm called PAGA, and it produces this embedding where we can sort of relatively clearly see this trajectory, which we can fit using an elastic principal graph. And we can see the trajectory. There's several branches here, but we sort of focused on this neurogenesis branch from olfactory stem cells to olfactory sensory neurons. But there are other branches, of course, producing sustentacular cells, which are supportive cells, ductal cells, and here this microvillus branch producing tuft cells and ionocytes, which I've been talking about quite a lot. Moving along the neurogenesis branch like this, we could then look along pseudotime. So these are the same cells, but ordered from stem cell to mature neuron. We could see that the marker protein, olfactory major protein or marker protein, I forgot, turns on towards the end as expected. And then we could look at all of the genes which really do turn on along this trajectory and identify both late markers of mature neurons, so here's OMP, but also early markers of committed precursors that are on their way to differentiation into neurons. And so we identified this new marker, Stathmin-1, as a new marker of committed precursors. If we visualize, so here on the x-axis is this pseudotime normalized between 0 and 100. So 0 is a basal stem cell right at the beginning, and 100 is an olfactory sensory neuron right at the end of differentiation. Here's K5, the stem cell marker, which turns off. P63, stem cell marker, which turns off. OMP, the neuron marker, which turns on. We could identify these new markers which turn on early in the trajectory, which is present while there are still K5 expression. So these are kind of stem-like cells, but they're not yet expressing markers of mature sensory neurons. So this is a sort of early marker. And then when Laura stains for Stathmin-1, we can see that it's I really love this stain. I think it's beautiful. It's really right where you would expect it to be, right after in terms of this differentiation sort of proceeds from the basal stem cell to the olfactory stem cell, to the olfactory neuron. Now our pseudotime is going in that direction. We can see that K5 is in the basal membrane as expected. OMP is in the luminal surface as expected, and Stathmin-1 is right in the middle, which is really, really beautiful. And it provides sort of validation that this is a new marker of neuroprogenitors, and also that they proliferate, that the proliferating cells that appear after alternaria are also positive for Stathmin-1. One of the things that we then really wanted to know is there's this established fact in the field, which is that the main driver of olfactory stem cell proliferation is actually tissue damage. So if the tissue is stripped by a chemical injury or some other damage, then the stem cell compartment really proliferates in order to regenerate it. So we wanted to see whether that was what was going on when we observed this stem cell proliferation. But this stain really showed that this is a stain of the olfactory mucosa after alternaria, that its architecture is remarkably preserved, and the thickness of the olfactory layer, as quantified here, with PBS and also after Alternaria really doesn't change. So there's no profound damage to the olfactory epithelium. And the next question was, of course, is there apoptosis? So we used the tunal assay. So we can see no apoptosis at baseline. After Alternaria, there is some apoptosis, but only in the respiratory epithelium. And the olfactory epithelium, which is marked by the presence of OMP for olfactory sensory neurons, there's really no apoptosis. So this induced proliferation of stem cells in the olfactory epithelium really occurs in the absence of tissue damage or apoptosis, which was really interesting. So then we wanted to see, okay, we already know about one mechanism by which allergens, or in this case, type 2 stimuli, so the worm, can trigger stem cell proliferation as part of a tissue response. This was what I talked about before in the gut, where tuft cells can trigger this response. So we wanted to know, like, are we seeing an analogous response in the nose where tuft cells trigger stem cell proliferation in a manner that's mediated by ILC2s and IL-13? And is it similar to the so-called weep and sweep? So we had these two potential cellular players, tuft cells and innate lymphocytes. And so Laura's group used two genetic knockout models to sort of test these hypotheses. The PO2F3 knockout mouse, which is deficient for tuft cells, and IL-7 receptor knockouts, which are deficient for lymphocytes, and STAT6 knockouts, which are deficient for IL-13 signaling. So we wanted to use these mouse models to sort of provide strong evidence of whether these cellular players are involved. So as expected, when we looked at eosinophilia, so the infiltration of these inflammatory immune cells into the nasal mucosa after the treatment of Alternaria, so on the x-axis, we can see sort of robust eosinophilia in wild-type mice, which is abolished in the IL-7 receptor and in the STAT6 knockouts. So clearly, lymphocytes and IL-13 are both required for eosinophilia. But what was surprising is that when we look using intracellular facts for the proportion of K5, Ki67 double positive proliferating HBCs, there's this robust increase in naive mice, which is actually preserved in the lymphocyte deficient and IL-13 deficient mice. So this induced proliferation actually doesn't depend on lymphocytes and IL-13. So we then did an RNA-seq experiment to test the same thing at the mRNA level. We saw the induction of Ki67 transcript and also a cell cycle signature score, which uses around 100 cell cycle associated transcripts. Both of these are robustly induced in wild-type mice by Alternaria, and this actually does persist in both the lymphocyte deficient and IL-13 deficient mouse models. So this HBC proliferation doesn't depend on these lymphocyte-driven or IL-13-driven circuits. So what about tuft cells? When we looked at eosinophilia, it was reduced in the tuft cell knockouts. So this is the orange and now PO2F3 tuft cell deficient mice. So eosinophilia was reduced. But what was really cool was that the induced proliferation of stem cells was actually really abolished. And we really don't see a significant increase in proliferating stem cells in the tuft cell deficient mice. So, and this was also true in our RNA-seq experiment. So there's both no increase in the proportion of proliferating stem cells by FACS and no increase in the expression of cell cycle transcripts in isolated HPCs by bulk RNA-seq, so no increase in MKS67 and no increase in the cell cycle signature score in the tuft cell deficient mice. So this was super interesting, and we performed a sort of comparative transcriptional analysis of these sorted HPCs. So now each column here is a pool of sorted horizontal basal cells, either from naive mice or exposed to Alternaria. So these are wild type at the top, this first section, and then these are the three knockout mouse models I've been talking about: tuft cell deficient, lymphocyte deficient, and IL-13 deficient mice. And we sort of isolated the genes which were upregulated by Alternaria, but knocked down by one of these knockout mice. And I won't go through this in super detail, but the sort of take-home message was that the genes which were reduced only in PO2F3, the genes which were really dependent on tuft cells, or the genes whose alternaria-induced expression really depends on tuft cells, included Ki67, TOP2A, and all these markers of the proliferation response. So it was clear that a tuft cell ablation really specifically blocks the induced proliferation of olfactory stem cells. So to sort of conclude, we've started to see that there's a tuft cell dependent neuroepithelial stem cell activation. So these basal cells really proliferate in response to an inhaled allergen. But unlike in the gut, this tuft cell dependent stem cell activation doesn't depend on IL-13 or lymphocytes. But I think what was really interesting to us is that since there's no cell death, there's really no need to produce a whole bunch of new epithelial cells. And it may be that this induced proliferation is actually bad, it's actually aberrant, and it may not be a good thing for the tissue. And one of the things that we've been thinking about from reading the literature, as and particularly Nora Barrett's recent study, is that these basal cells are actually really enriched in nasal polyps, which are a feature of chronic rhinosinusitis, as you all likely know better than me. And so it would be very interesting if these tuft cells were signaling to the olfactory stem cell and driving pathological proliferation. So one of the take-home messages is that tuft cells have been established now for a few years as drivers of type 2 inflammation. But now these new findings are implicating them in injury repair, regeneration, and potentially in tissue pathology. To summarize some of the findings that we were able to show across the nasal mucosa, we've identified that there are these new subtypes of tuft cells called tuft MVC or tuft microvillus cell. This is the one with the globular morphology. And that these two types of tuft cells, these two types of microvillus cells actually correspond in the olfactory epithelium to tuft cells and to ionocytes. And I didn't actually really talk about this at all, but we identified that the third subset of tuft cells is resident in the lateral nasal gland, and we term this one tuft gland, which has its own unique transcriptional profile. And so for more details, all this data is not published, but it is on bioRxiv. So you can go to this URL, tinyurl/nasal tuft cells to read more details. And I want to thank everyone who was able to contribute to make these studies possible. From my group, Evan Lemire, a really talented PhD student who led a lot of this work. Wenjun Deng and Nicole similarly contributed, and Laura's group was really instrumental in co-leading a lot of these studies with me, and particularly her postdoc, Saltanat. And I also want to thank Josh Boyce, who provided LTC phosynthase mice, and Tanya for access to human samples, and Nora Barrett's group for some techniques to help isolate these basal stem cells. So thanks so much. Here's the link to the preprint again, and let me know if you have any questions.
Thank you very much, Adam, for novel insights into what we thought we knew about, and now we're learning so much more about airway mucosa, thanks to your work and this exciting collaboration with the allergy group. I'm sorry for the rough start to our program today. There was a question submitted that had to do with the cellular proliferation that you observed in mice when exposed to allergen. And I think you may have answered this question, but the question came in, did you see these responses in naive mice rather than those who had been, I guess, conditioned for allergen? If so, what might the mechanism be by which an allergen stimulates these cells?
Yeah, it's a good question. We didn't observe this proliferation when there was no allergen present. It was we only saw it after the. I didn't describe the allergen time course at all, but there's a sensitization step and then a challenge with alternaria. but it happens quickly. We're looking only at the innate response. So it's not like a 2-week challenge. It's like within a few days. So it's a 24 or 36-hour treatment. And so, yeah, we're not actually sure what the mechanism is. We looked at whether Alternaria itself can directly damage stem cells in culture, and it doesn't look like it can. So it basically appears that there are molecules on these tough cells which are able to sense proteases in the allergen and secrete some signaling molecule which act on the stem cell. That's our hypothesis right now, but we haven't identified either the receptor on tough cells or the signaling molecule which activates the stem cell, and that's sort of the focus of our current work.
It's always been this sense of analogy between parasitic infections and active immunity and allergen, and I wonder if you see any similarities between reaction to parasites and reaction to allergen.
Yeah, certainly. I mean, I agree with the analogy. I haven't studied an allergen and a parasite in the same system. The work I presented in the intestinal epithelium was studying with a parasite, and we did use the H. polygyrus model as a type 2 model. And in the airway, we looked only at allergens. So I haven't actually looked at like a head-to-head comparison of which pathways are activated by the allergen and which by the parasite. I think there's a sort of tendency to assume that they'll be the same, but likely that's not the case. And there will be aspects of the anti-parasitic response which are recapitulated by the allergen, but there'll probably be aspects which are not. I actually don't know of any studies that have compared them. It might be an interesting thing to try to do.
Is someone interested in cough and cough reflexes? Is this sort of approach to identifying cellular constituents potentially applicable to the cells in the airways related to cough and their proliferation in people with cough, etc.?
Yeah, absolutely. I mean, it's not quite my area, but I know that like a lot of those reflexes are mediated by nerves, basically, which innervate the airway, like the sensory nerves and also parasympathetic nerves and motor neurons, which govern the cough. And there are groups at Harvard Medical School, particularly Steve Lieberlis's group, which is sort of, but also Isaac Chu's group, which are sort of mapping out the different populations of nerves which innervate the lungs and which drive some of these responses, including cough. And single-cell-seq is now a kind of like really important tool in everybody's, certainly in those groups' arsenal, to sort of map out which cell types are driving cough, or which are driving particular airway reflexes. But they certainly had, Steve Lieberlis's group certainly had a beautiful study in Cell a few years ago, in 2020, I think, showing that swallowing was mediated by a particular rare subset of airway innervating neurons. So I don't think they've yet identified like a rare one that mediates cough, but maybe that's like in the works.
More nerve mediated than cell mediated, maybe.
Yeah, I think so. Yeah.
Right. Well, thank you very much for a terrific presentation. Well done. And thank you all for joining today. We'll start up again asthma grand rounds in the fall. Take care. Good summer.
Thanks so much.
Thank you.